welcome to our first Irish Embassy members talk. And I'm going to be talking about the human microbiome and try to convince you that it is really an essential physiological requirement for humans. I think you'll be pretty impressed. It's almost impossible to pick up a journal without finding some kind of paper on the microbiome. Here you see some prominent journals where the entire issue was devoted to the microbiome. So the problem in trying to cover this kind of a topic is that there's so much. We could be talking for days, and I know you don't want to do that. So what I've done is I've tried to select certain studies, research papers that'll give you an idea of the incredible interactions between the microbiome and humans. The NIH has the Human Microbiome Project, where they're trying to identify all the different little bugs that live in our bellies. It's a major project, easily equivalent to the Human Genome Project. So there are a lot of them, about 100 trillion, so that's a lot of zeros, 10 to the 14th. And the human's body only has 10 to the 13th cells. So you have, for every human cell in your body, there are 10 bacterial cells in your body. You have more bacterial DNA than human DNA. The human genome has, 23,000 genes. The microbiome has over 1 million. So just kind of think about these numbers. They're pretty amazing. Holy cow. So here you see sort of a close-up of some microbes in the gut, right on the gut epithelium. So what does it do? And I also want to cover prebiotics and probiotics. I'm sure you've all heard of those things. So one thing is that they ferment undigested food. All the food that you eat is not completely digested and absorbed. And what's not gets down into the colon where these bacteria live, and they ferment that. And then some of these also synthesize nutrients. A lot of the vitamin B12 is not from your diet. It's synthesized by bacteria in your gut and then absorbed. And then some of the beneficial bacteria produce, which are usually lactic bacteria, they produce short-chain fatty acids like acetate, butyrate, propionate. And these short-chain fatty acids are really important fuel sources for the gut epithelial cells. And because they're acids, they reduce the pH. Well, pathogens and potential pathogens like E. coli, staph, strep, they don't really do well in an acid environment. So by producing these short-chain fatty acids, these beneficial bacteria suppress the growth of those potential pathogenic species, and then they can predominate in your gut. They also interact directly with gut epithelial cells to help them mature, and they interact with the immune system. 70% of your immune system is the GI tract. 70% of the immune cells in your body are in your GI tract. It is the largest immune organ in the body. And these microbes play a critical role. So to show that, this is one of my favorite research models. It's called the germ-free mouse. I know that sounds like an oxymoron. So the germ-free mouse is a mouse that's delivered by a C-section. You can see the little baby mice in the back there, a very tiny C-section. And they're delivered in a sterile environment. So they have no bacteria in their gut. They are literally germ-free. So I'm going to show you some micrographs taken from these mice. So this is from a normal mouse that is normally colonized with bacteria. So the slide on the left, those are T and B cells from the spleen. All that red dye, those are T and B cells in the spleen. And remember, the spleen is remote from the GI tract. The middle picture are T and B cells in the intestinal wall, and the one on the right, those are That's IgA and immunoglobulin and Peyer's patches, which is a part of the gut immune system. So that's a colonized mouse. This is a germ-free mouse. There's not a lot of red there. In other words, if you don't have any bacteria in your gut, there's very poor immunological development. It's absolutely required for normal development of the immune system. And another way of looking at is antibiotic-induced dysbiosis. I love these words. Dysbiosis is an imbalance in the microbiome. So this is a study that was done to see how antibiotic exposure during the first year of life affected the risk of developing food allergies. Pretty large study, almost 6,000 babies, and 1,500 of them developed allergy. And looking at the number of prescriptions for antibiotics in the first year of life, perhaps not surprising, but also kind of shocking. Even one prescription in the first year of life, that's a 1.25 increased risk of developing food allergy. 5 or more, that's a 1.65 increased risk. And when you think about the number of antibiotics that are routinely prescribed to children, is it a surprise that we're seeing so much allergy, asthma, immune-related dysfunctions? And you can see it in the global antibiotic consumption. This is the billions of daily doses from 2000 and from 2015, 65% increase. The other way you can look at it as billions of daily doses per 1,000 inhabitants, and that's also gone up dramatically. And most of that is in low-income countries. So here's the increase in low and middle-income countries, and this is high-income countries. Because in a lot of these countries, you don't really need a prescription. You could just go to the drugstore and say, I'd like some penicillin or erythromycin. And in terms of daily doses per 1,000 inhabitants, it's actually gone down in high and middle-income countries, while it's gone up dramatically in those low-income countries. And take a look at poor preterm babies, early antibiotic use in premature infants. It's almost all of them are getting antibiotics. Yes?
So is that prescribed antibiotics, or does that also include...
That includes unprescribed. So that's just antibiotic use.
But I mean, like, is that antibiotics in food?
No.
Or in meat?
No. These are medicinal antibiotics, pills. But, that's a good point, because, George down the street, he gets a prescription for erythromycin, he gets halfway through it, and I feel pretty good, so he flushes the rest down the sink. Well, now it's in your drinking water. So there's no question that our environment is contaminated with antibiotics, and they have a profound effect on our microbiome, as you've just seen. So this is Clostridium difficile. First gained attention back in the 70s when some crazy doctor said, well, I think ulcers are actually caused by bacteria. And 20 years later, he was proven right. Clostridium difficile is a primary cause of ulcers, but it also causes something called ulcerative colitis. This is what a normal colon looks like. You're not freaked out by seeing body parts, are you? No, good. So, nice and pink and healthy looking. This is what it looks like when you have ulcerative colitis. So it's a really nasty disease, and especially in the elderly, it can be not only devastating, but completely resistant to normal antibiotic treatment. Thus comes the idea of fecal transplants. So this came up originally from just a regular practitioner in Michigan, and he was talking to a friend of his who was a vet, and he was describing a patient with ulcerative colitis that wasn't responding to anything. And the vet says, well, when horses have colitis, we feed them manure from other horses and it seems to work. So he says, well, gee, that sounds like an interesting idea. This is a study he did. So these are 18 patients, about 72 years of age, and they had chronic, untreatable C. diff ulcerative colitis. So this is how the fecal transplant works. They collect feces from a family member or someone living in the household. They have to be healthy. They have to be free of any antibiotic use. And they then dilute the feces in solution and then give that through a nasogastric tube. So you're not drinking it, so don't worry about that. It goes directly down into the stomach. So they're actually inoculating these patients with fecal material from a healthy individual. And this is the outcome from the study. So there were a couple patients who died from kidney failure, pneumonia, probably not related to the ulcerative colitis. One patient failed. 15 of them were cured. That's an 83% cure rate. You don't see that with antibiotics. So this is remarkable, and now they are routinely doing this in a lot of big institutions like Mayo's, and there are now banks, fecal banks, and they search out for people who have healthy shit. So, and they're really scrupulous about looking and making sure that you haven't been using any antibiotics and that you have a good meal. Pretty fascinating. Roseomonas mucosa. This is what it looks like on a petri dish. And this was isolated from skin. So yes, we have microbes on our skin too. They're all over. So this group was interested in eczema, atopic dermatitis. So these are people who have really severe skin disease. So they started by looking at how pathogens would grow in the presence of this skin bacteria. So what you see here, the HV, that's our mucosa collected from the skin of healthy volunteers, and AD is the same bacteria collected from the skin of people who had atopic dermatitis or eczema. And it's compared to a control, which is just a petri dish without anything added to it. So when they're grown in the presence of our mucosa from healthy volunteers, it actually suppresses the growth. This is Staph aureus, which is a nasty pathogen. And when it's grown in the presence of that same bacteria from people with atopic dermatitis, it actually increases the growth. So the same bacteria from the skin of healthy people suppresses the growth of a pathogen, and that same bacteria from the skin of people with eczema stimulates that pathogen. So then they tried it on a mouse. So this is a mouse model of atopic dermatitis. When they treated that mouse with the bacteria from healthy volunteers, you see that pretty good looking mouse here. This is the mouse that was treated with the same bacteria from the skin with atopic dermatitis. Well, after that, they thought, well, maybe we should try this on people. And they did. So this is a study where they collected the same bacteria from the skin of healthy people and used it to treat patients who had eczema. The anticubital, that's the inside of your elbow, which is a real common place that eczema shows up. So the scorecard is a way they rate the severity of eczema. And you can see that on the left is their pre-treatment score, and on the right, their post-treatment score. So that was an average, almost 60% decrease in the score. This is looking at itching, pruritus, and there too, a dramatic decrease. almost an 80% decrease in itching. And look at that, 5 of these patients went to 0 itching. Now I'm sure you want to see what it actually looks like. So this is the antecubital elbow pre-enrollment. And this is after 6 weeks of treatment, and this is after 10 weeks of treatment. In addition, all the subjects in the study were allowed to use the same treatment in other parts of the body where they might have something showing up. So this is a subject, you can see that's pretty severe eczema, that's pre-treatment, and here you see 6 weeks later. Is that not amazing? So this is just being treated by a bacteria that's found on our skin, unless you use antibacterial soap, which might not be a good idea. But there are a number of other conditions that are pretty interesting connected to the microbiome. So I'm going to talk about a couple of them: obesity and non-alcoholic fatty liver disease. So this is when there's fatty intrusions into the liver not related to alcoholism. This is just you and I. And this is pretty prevalent. It's the most common liver disease. It's present in 30% of the population. Look around you. 30% of the population has non-alcoholic fatty liver disease. So this is the study that shows this relationship. So this is a strain of mice, and they took this mouse strain and they gave it a high-fat diet. And then some of the mice on that diet they called responders, and those they both gained the same amount of weight on this diet. So there was no difference in weight gain, but the responders became hyperglycemic, high blood sugar, and they also had increased levels of inflammatory cytokines. And the non-responders were normal in blood glucose and these inflammatory cytokines. So by doing this, they essentially identified unique individuals within the same strain of mice that had different responses to the same diet. Then they did an experiment where they had 2 groups of the same strain of mice, but they were germ-free. Do you remember what the germ-free mouse is? I love germ-free mice. And they were both raised on a low-fat mouse chow diet, and then they were colonized with bacteria from either the responders, the ones that developed problems, or from the non-responders. So this one strain of germ-free mice, when they were colonized with bacteria from the responders, and they were both fed a high-fat diet, on that diet the responders became hyperglycemic, hyperinsulinemic, that means high levels of insulin, and they developed fatty liver. The same strain of germ-free mouse inoculated with bacteria from the non-responders, they had normal levels. Here's what their livers looked like. So this is a micrograph of the mice that were colonized from non-responders and here from responders. And all that white area, that's fat deposited in the liver. Our livers are pretty important. It's often referred to as the brain of the body. It's the most metabolically active organ in the body, and it's a major control of much of our metabolism. Discordant twins. So these are identical twins, and in this case, they're discordant in terms of obesity. So one of the twins became obese, and one remained lean. So they're identical twins, genetically identical. Then they collected a fecal sample from each of these twins and used that to inoculate our favorite little model here, the germ-free mouse. Well, gee, what do you think happened? Yes, it did happen. So this is the percent change in body composition. So the germ-free mouse that were inoculated by the lean donor had almost no change in lean body mass on the obese side. So lean body mass is everything in your body that is not fat. So it really is not just muscle, it's also bone, organs, literally anything that's not fat. And then the fat mass, a dramatic increase. And when you look at the percent of increase in fat mass, virtually none in the mice that were inoculated from the feces of the lean woman. So how is it? What are the mechanisms? How is it that the microbiome can affect things like metabolic syndrome? Well, this is one theory. And as I said, a lot of these microbes produce short-chain fatty acids. Well, if you have dysbiosis, that imbalance in the microbiome, then you may have an overproduction, in this example, acetate. And that acetate gets to the brain and it affects parasympathetic activity. It's part of the nervous system. And that affects things like insulin release. Garrulin is the hormone that stimulates our appetite. So if you have a lot of garrulin, you're hungry all the time. And that, of course, increases nutrient intake. And as you increase nutrient intake, you get feedback because that kind of stimulates that whole process. And as a result of that, then you get these consequences, obesity and metabolic syndrome. So this is one of the ways in which our microbiome can affect our whole metabolism. How do we get our microbiome? Well, infants are inoculated at birth. So it depends on the method of birth. If it's a normal birth, they're inoculated by the bacteria in the vaginal canal. I always wonder about these water births. Is that chlorinated water? I wonder what that does. And then, of course, what hygienic measures are in place? What's on the surfaces in the birthing area? Is it a hospital? Is it a home? Any of these surfaces could be influencing it. Antibiotic use. In this country, women are routinely tested for group B stress before delivery, and if they're positive, they're given antibiotics automatically because they don't want the infant to be infected. Well, what's that do to the bacteria that the infant is inoculated with? And of course, the type of feeding can have an effect. But the method of birth is kind of interesting because we have a lot of infants that are born by C-section. So there were hospitals that were taking vaginal swabs for infants born with C-sections and swabbing down the infant, thinking, well, at least they'll get exposed to that vaginal bacteria. Well, the American Academy of Gynecology came out with a really strongly worded statement, said, don't do that. You don't know what you're inoculating them with. It could be pathogens. So it was interesting that they took that very firm stand. And since then, I've seen papers where they compared the microbiome of infants delivered vaginally by C-section showing no difference. And then I see another paper showing there are differences. It's very difficult to tease this out. They're probably both right. There are probably some cases where there aren't differences. And oh, what about pets? Oh yes, that has an effect too. I just came across this paper today. 2017, exposure to furry pets influences the microbiome. Is that good? Is that bad? Probably good. Probably good. Get those pets. So initially it was bifidobacteria and lactobacilli that were identified as the beneficial bacteria. Now there are a lot of different species, but I first made this slide, I think, in the early 90s, and you can see the number of bifidobacteria and the number of lactobacilli that were identified at that time. Well, this is 2016, bifidobacteria. This is 2016, lactobacilli. See, we used to identify these bacteria by growing them on plates and then looking at the morphology, looking at their shape. Well, now it's DNA. So it's a much more specific, much more precise identification. The number of species is remarkable, and this is just within lactobacilli. And when you look at sort of the phylogeny of the microbiome, this isn't even getting close to that level. And I can assure you that there are many more species that have not even been discovered yet. And it's all happening right inside your body and on your skin. We know that breastfed infants tend to have higher levels of bifidobacteria and lactobacilli. Formula-fed infants tend to have more adult-like bacteria. So this is a feeding effect, starting with infancy. Probiotics are live microorganisms that you take orally to have a health benefit. And you've all seen probiotics in the stores, so you know what they are. To be, and then of course when the skin microbiome came out, now we have a whole book of dirty skin and how important that is. And of course, now there are probiotic-infused skincare products. So if you want to wipe a little lactobacilli on yourself, you can find something to do that. So in order to be effective, a probiotic, first it has to be free of transmissible antibiotic-resistant genes. Those little strings you see between those bacteria there, they're sharing genes. So if you have a bacteria in your, especially if you're taking a probiotic and it has a gene for antibiotic resistance that it's going to transfer around in your microbiome, that would not be a good thing. The other thing you need to know is that it remains viable during manufacture. What's the manufacturing process? Do they test whether what you're taking is actually alive? And it has to survive digestion and get down to the colon, still being viable so that it can have an effect. So when you say that something is colonized, that means it's a permanently living in your gut. It's a colony. No one has ever shown that probiotics colonize, which is, it's almost counterintuitive, but when you're taking probiotics, you can detect them in the stools. When you stop taking them, you can't detect them anymore. That doesn't mean that while they're there, they're not having those beneficial effects, but you have to take them routinely in order to have that benefit. They don't become permanent members of your microbiome. But probiotics, diarrhea, traveler's diarrhea, if you're traveling to another country, take probiotics with you, but it's also been associated with H. pylori infection, irritable bowel syndrome, vaginal bacterial infections, cold and flu. But there's some pretty interesting studies. These are some of the common species, but there are really a lot more. But Lactobacillus bulgaricus and Streptococcus thermophilus, that was the original yogurt. Bulgaricus, Bulgaria, that's where yogurt came from. And then when Actinia first came out, they used Bifidobacteria animalis, but they renamed it Bifidus regularis and patented the name. A lot of marketing involved here. Yoplait used B. lactis. Stonyfield Farms, L. casei and L. reuteri. These are two patented bacteria. So when you identify a bacteria by DNA analysis and no one ever has reported that, you can patent that. So yes, there are patented bacteria. This is a study done with probiotics in older women to see if it would affect bone loss, osteoporosis. They were doing bone mineral density tests in order to test that. This is a double-blind, placebo-controlled study. So when you look at research, it's important to look at the design of the study. In order to have confidence in the outcome, you need to know that the researchers and the subjects are completely blind to what they're getting so that their knowledge doesn't bias the outcome. And that's what double blind means. Double blind means neither the researchers nor the subjects know what they're taking. Placebo-controlled means the only difference between the two groups is one factor, so that any differences you see, you can be confident is because of that one factor. If you have a lot of differences, you don't know what's causing the effect. So you have to control that as much as possible. And you can't control everything. So there are a lot of uncontrolled effects that you don't even know about. The only way you can control for that is through randomization. So you randomly select subjects to each group, and the hope is that by just randomly selecting, those uncontrolled factors will be equally distributed between the two groups. But you can have bad luck. So you may have a randomization and you still might end up with one group, all who has one confounding factor. So when we were doing studies with infants, you collect the data on smoking in the household, number of siblings, attendance at daycare centers, and you can look at that statistically and see if it had an effect. And if it does, you can put that in your statistical model. But you can't test for what you don't know about. So when you look at these studies, double-blinded, randomized, placebo-controlled, that's what you want. This is the lower leg, and the fibula is the smaller bone on the left, the tibia, the larger one, and they were looking at the end of it, the distal tibia, because it's a place where elderly women commonly show osteoporosis. And they were also looking at what they call the trabecular bone, which is that sort of grainy, coarse inside of the bone. And then they randomized them to get either a probiotic, Lactobacillus reuteri, or placebo. And they're, to be truly blinded, you can't tell, you should not be able to tell the difference. These are capsules, so that's pretty easy to do. So this is looking at the bone mineral density in the distal tibia. So they all lost bone over the course of the study, but the ones who were on the placebo lost significantly more bone. Here are the outcomes for the lumbar spine and for the total hip. And although they look like they're equally impressive, they weren't statistically different. So what that tells you is there's a huge amount of variation in the response. And that's why we don't teach statistics in school, certainly not through high school. And when you're trying to understand studies like this, the only way you know what it means is if you understand a little bit. So when you see a p-value of less than 0.05, that means you're 95% confident that this is a real difference. And that's what is generally the level that's considered proof. And then with trabecular bone, that was also a significant difference. So the only difference between these two groups of women is that one was getting a probiotic, L. reuteri, and the other was not. So for microflora to grow and provide these benefits, obviously they have to have food. And then they ferment that food, that undigested food, it produces short-chain fatty acids and then the decreased pH and all those benefits. Well, what if we provide the food? That is what we call a prebiotic. A prebiotic is very specifically defined as a non-digestible carbohydrate. It's not digested by human enzymes. It travels through the gut and it gets down to the colon where it can only be fermented by beneficial bacteria. So prebiotics cannot be used by E. coli, staph, strep, clostridia, but only by the beneficial bacteria. So there aren't a lot of clearly defined prebiotics. Human milk does have prebiotics. Human milk oligosaccharides. These are a group of sugars. They're oligosaccharides. They're synthesized from lactose. They're genetically determined. They're actually related to blood type. And it's a major component of human milk, and they act as prebiotics. They are preferentially fermented by beneficial bacteria. So if you look at the older literature, when you're looking at the composition of human milk, you'll see something called bifidus factor. Whenever you see the word factor, that means they don't really know what it is. No, seriously, that's what it literally means. So bifidus factor, we now know, are these oligosaccharides. So they knew there was something about human milk that supported the growth of beneficial bacteria. They just didn't know what it was then. Then there are plant-derived. Inulin is the most common. The richest source, your favorite vegetable, the Jerusalem artichoke. I know Sarah's eaten them before. I don't think I've ever had one. Chicory is also rich in inulin, but it's found in a lot of fruits and vegetables. And of course, now you can buy it as a supplement, usually derived from the Jerusalem artichoke. And again, human milk. Oligosaccharides, there are more oligosaccharides in breast milk than protein. It's the third largest component of human milk. So that gives you a sense of physiologically how important this is, literally from birth. There are also synthesized galacto-oligosaccharides and fructo-oligosaccharides. These have been added to infant formula since the late 80s, at least in Japan, in the US more around the 2000s. And lactulose, which is clinically used to treat encephalitis because of its effect on the microbiome and how much ammonia gets into the blood to keep it from getting to the brain. Now, if you go crazy with prebiotics, remember they are fermented. And when you get all that fermentation in the gut, go back to high school science, remember osmotic pressure? It pulls water into the gut and causes diarrhea. So, you know, people who see the sugar-free candy and it has those sugar alcohols, those are also non-digestible. Oh, sugar-free, I think I'll eat half a bag, and then they're on the pot the rest of the day because they got severe diarrhea. So they're like with anything, there are consequences to overindulgence. And inulin is now being added to a lot of commercial products. Here you see cereal, Crystal Light, it's in bread. I've seen it in dog food. But with most of these, if you see on the label contains prebiotics, look at the ingredients to see what they've actually added. The nutrient label doesn't really tell you what you're eating. You have to look at the ingredients. Do you look at the ingredients of what you eat? I would expect that at this group. And then you see these types of claims. Prebiotics protects you and supports the growth of the good bacteria already in your body, which is accurate. It's not unreasonable. This is a study looking at the effect of prebiotics on the bacterial population in infants. So these are infants fed formula with or without prebiotics. These are the levels of bifidobacteria after 3 and 6 months in breastfed infants. Here in infants fed a formula containing galacto-oligosaccharides, the prebiotic, and here a control without. And those are all, the control is significantly lower than the other two groups. This is looking at the effect of prebiotics on secretory IgA. IgA is an immunoglobulin. Secretory IgA is IgA that has a little protein attached to it, secretory protein, and it's secreted into the gut into the lungs, tears in your eyes, it would be the primary defense against infectious diarrhea, for example. So this is the secretory Ig level in breastfed infants, infants fed a control infant formula, and those with a prebiotic formula. Look at that breastfed group. Does anyone know why it's so high with those breastfed infants? It's because breast milk is loaded with secretory IgA. It's one of the main protective benefits of breast milk. The immune system of the breastfeeding mother surveys her environment, identifies pathogens in the environment, and then synthesizes pathogen-specific IgA for her breast milk, so her infant is protected against precisely the pathogens it's most likely to encounter. So at 26 weeks, after the breast milk IgA has basically dissipated, you see that the prebiotic group had the same levels as the breastfed group, and significantly higher than the infants on the control formula. So just from looking at that effect on IgA, we see at least one indicator that prebiotics stimulate the human immune system. Well, can they change a clinical outcome? This is a study where infants who were exclusively formula fed were randomized to get formula with or without prebiotics for 6 months, and they collected data for 2 years on the incidence of allergy and infections. This is looking at allergy, and that's atopic dermatitis, eczema, wheezing, and allergic urticaria. These are the control infants, and these are the infants who got the prebiotic, all significantly lower. And also in terms of incidence of disease, overall infections, upper respiratory tract infections, infections that are treated with antibiotics, and episodes of fever, control and the prebiotic group, all significantly lower. So this is just by adding what's basically a non-digestible sugar to the formula of these infants. And then this is another recent study. These are juvenile rats fed a diet rich in prebiotics and what they called bioactive milk fractions. Don't really know what that was specifically, but you can see that it altered gene expression in the brain of these rats, gene expression in the brain, and it affected their emotions. Yes, rats do have emotions. I know it's hard to believe that, but rats, when they have a lot of angst, they get really nasty. They bite. I mean, there are really obvious changes, social changes. So what they're saying is this diet fed early in life can alter brain plasticity and behavior. Now again, this is a rat study, but there are some interesting human studies. So back to human milk, and again, the human milk oligosaccharide fraction, you can see it's more than protein. These are the major human milk oligosaccharides. 2-fucosyl lactose. Don't you love these words? They just kind of roll off the tongue. And so we'll just call it 2FL. It's a lot easier to say. And I'm going to show you data on this because it's so remarkable what a simple molecule can do. So the 2FL, it's a trisaccharide. So it's made up of glucose, galactose, and fucose. These are all simple sugars. So keep that in mind as you look at this data. And because of the structure, it is non-digestible. So it's not digested by human enzymes, and it does act as a prebiotic. So I'm just going to show you a couple interesting pieces. And the company I used to work for, Abbott, recently added this to infant formula, which is why I have all this data. But it's when bacteria evolved, they learned to recognize receptors on the gut epithelial cells. And when they attach to those receptors, that's how they cause disease. That's how they cause an infection. Well, these human milk oligosaccharides imitate those receptors. So the bacteria binds to the oligosaccharide instead of the receptor, and that way can prevent an infection. So this is all beyond simply acting as a prebiotic. And there are what we call in vitro data to support this. So this is a model of the gut epithelium cell. They take transformed gut epithelial cells and they grow them in a petri dish so that the whole petri dish is covered with a layer of these cells. They're called CO2 cells. And they use that as a model to determine mechanisms for nutrient absorption or infection. It's a very classic model for looking at gut function. In this study, they were looking at the adhesion. The Campylobacter jejuni is another one of those nasty pathogens. When it attaches to the gut epithelial cells, it causes an infection. In this study, they were looking at how different substances would interfere with that attachment. So this is lactose. You can see there was 100% attachment. It had no effect at all. But 2FL, like human milk, led to a significant reduction in attachment. So this is how you use in vitro data to support a scientific concept. So it's a lab study, and it is meant to mimic what's happening in our bodies. Whenever you add a novel ingredient to an infant formula, the FDA requires that you do a growth study, and Abbott did this study. But during the study, physicians who were seeing the infants in the study recorded all the cases of eczema that occurred in the study, and there was a significant difference. This is the control group, the human milk group, and the 2FL group. Now, this was the other thing you need to look at when you do a study. All studies have what's called a primary outcome variable. So the researchers have to pick one thing that they're looking at and design the study to see that one thing. They collect a lot of other data, but say if you're doing a drug study for the FDA, they will only look at the primary outcome variable. They don't accept anything else. So this was not a primary outcome variable, so this data certainly couldn't be used to say 2FL prevents eczema. You'd have to do another study where that is the primary outcome variable. But nevertheless, kind of remarkable. And I could spend 15 minutes showing you animal studies that support this data, but I'll spare you that. This is a really remarkable study. Necrotizing enterocolitis, NEC. It's the most feared disease for premature infants. The gut becomes necrotic, it dies. And it's a horrific disease. It has multiple etiologies, so it can be a bacterial infection, but when it occurs, it's just devastating. For one thing, it often leads to gut resection. You know, we're talking about babies that weigh 800 grams having part of their gut removed, and they can't be fed, and so they're not growing. And when premature infants don't grow, their brains don't grow. So it's a devastating disease. Of course, there's a lot of research looking at NEC. So this is an experimental model of necrotizing enterocolitis. And this paper cracked me up. This is the control group. They said they were fed breast milk. Well, no, actually it was mouse milk. I guess they don't realize that mice don't have breasts. But the upper panel, you see a picture of the gut, nice and pink, healthy looking. The bottom is a micrograph of the villi. You see they're nice and intact. To induce NEC in baby mice, they fed them a formula. It was half infant formula and half canine formula. I guess something for puppies who can't nurse. They raised them in a hypoxic environment. So that's a very low oxygen level, an abnormally low oxygen level. And just to be sure, they fed him a pathogen. And you can see it was very effective. So that black area, that's completely necrotized gut. It is dead. And the villi you see are completely disrupted. The next picture I'm going to show you is of mice who were given that exact same treatment, except their formula had 2FL in it. A little trisaccharide. And what I like about this paper is they asked the question, well, what's it doing? How can it do that? And they looked at circulation to the microvilli in the gut. So here you see a dye showing circulation in the mouse milk-fed baby mice, those that got the NEC treatment and those that got the formula for 2FL, two different types of dyes. So although NEC has multiple etiologies, the common insult is something that disrupts blood flow to the gut mucosa, which leads to the death of those cells. This little trisaccharide, by stimulating blood flow to the microvilli, has that profound of an effect, which is pretty amazing. When you show that study to neonatologists, their eyes just pop out of their heads, because to be able to prevent NEC, that is the gold star of neonatology. This is more on 2FL, showing that this simple little trisaccharide has effects on learning and memory in rats. Rodents are really great for research. This is showing you the percent of rats that achieve a learning criteria. In this case, it was pushing a lever to get a reward of food. And they tested them in two different trials separated by 6 days. And this is not 2FL injected, this is fed to them through the GI tract. And so the rats who got the 2FL significantly more learned the criteria, and by the second trial, they learned faster than they did the first time. Well, this group of researchers, they're in Spain, followed that up with a remarkable set of studies. And to answer that question, they implanted an electrode in the brain of rats, in the hippocampus. The hippocampus is the part of the brain where you turn experience into memory, learning. And they stimulated it with what was called a high-frequency stimulation, and then measured the amplitude, how high the response was to that stimulation. So these are, and again, this is from being fed 2FL, not injected, so it's going into the GI tract. These are the control and the 2FL groups, and here's their response. So the rats who were fed the 2FL had a significantly higher and dramatically higher response. They followed that up with, this is one of the most elegant studies I've seen, pretty remarkable. They had a series of stimulations separated by a day, so on 2 different days. So these are rats that were fed 2FL. This is the baseline, and here you see the response, the high response. Then in a second group, they gave those rats a vagotomy. The vagus nerve is the nerve that innervates the entire GI tract. They cut that nerve. So this is the baseline for the rats that had the vagotomy, and this is their response. All significantly different and disappeared. What that tells us is that this simple trisaccharide is having an effect on brain function, brain learning, by interacting with nerve endings of the vagus nerve in the gut. Pretty amazing, right? And this is a beautifully designed experiment. So yes, a vagotomy canceled that effect. And there are a lot of neurodegenerative diseases that are now associated with the microbiome. And some of this research is ongoing, a lot of it is animal research, but you can see that there are mechanisms by which what's happening in our gut can affect our brain. Well, we've now seen that the microbiome has effects on multiple aspects of our physiology, our immune system, our brain, our overall health, gut health, oral health, all those things. And we've seen that there are connections between the gut and the brain. There are connections between the microbiome and the maturation of the gut, and between the microbiome and the immune system. But what we're really learning now is that these all interact together in multiple ways. One of the faults, I should say, of science is to look at systems as if they're independent and standing alone. They are not. They are intimately connected in multiple ways, and you see from this research how they affect each other. You can't look at a system independently and understand everything that's going on. The major diseases of our society, metabolic disease, obesity, high blood pressure, type 2 diabetes, these are intimately affected by the microbiome. So we call the liver the brain of the body, but is not the human microbiome truly a metabolic organ? It acts like an organ. They're not just a bunch of nasty little bacteria, it's a part of us. And I leave you with a couple of interesting questions. What does living in space have to do with the microbiome? Well, NASA has a project to look at that. And although we've been talking about the microbiome, what about the virome? What about all the viruses that live in our body? What are they doing? And if you think there are a lot of bacteria, think about viruses, and they do have effects. They can affect things like chronic viral infections, acute viral infections, novel viruses, like, Ebola. And then there are what are called bacteriophages. Those are viruses that live in bacteria. And there may be some that carry genes for antibiotic resistance. There may be some that are commensal, beneficial. And then there are retroviruses. We have viral DNA in our genome. Ooh. Kind of crazy. So whenever I start digging into these things, what's always most remarkable is, again, the more you know, the more you realize you have no idea. But yes, the microbiome is just another one of your organs. And if you want more information, this is a really good, informative website. Well, thank you. Hope you enjoyed it.